So welcome all. So this is already the 4th international conference, which is organized by our group. Our group is from NOVAhealth, Chronic Disease and Infection. And the group aims to promote the research and awareness, and also to foster the academic and the social discussions around this theme of chronic disease and infection. And that's why we are here today, because this is one of our activities to foster these discussions. So today, as you know, the conference is focused on microbiota and health. And we have gathered a very interesting and very important discussions that we hope that we will have with you, that we have gathered experts on this area, on microbiota and the health, and to provide you insights which are around the composition of a microbiome, how it is associated or influenced by nutrition, associated with infections, autoimmune diseases, metabolism, and also how can it influence the public health and antimicrobial stewardship. So thank you for joining us. I will just briefly go through the website to show you some particularities about this today's program, and then I will move on to the some note from our vice rector, Professor José Fragata. So just for you to know, we are also being recorded. And at the same time, this meeting is also being streamed by the NOVA YouTube. Right? So, and soon my colleague will also share with you the link for the YouTube, in case you want to join us through there. Right? So this is our nice website. I hope you are all seeing this. So as you see here, this is the organizing committee, and you can go through a little bit, go down and look for the program. And also you can see a little bit about our speakers and also the e-posters. If you click here, we have the posters already online. Right? And very important, if you want to join us on chat, I actually would like to really ask you to foment these discussions among us, because we are virtually meeting, but we wanted to make it as more interesting in terms of networking. We have this networking chat. You click here, it will direct you to the Telegram program, which is also a very nice way for us to share connections, contacts, and ideas. In my case, it has been working better through the cell phone, but it might also work through the computer. At the same time, as you know, that the Zoom has also a very nice chat. So if you want to ask or to expose some of your opinions, you can please use the chat. Right? So this during the discussions, the moderators will use the chat to conduct the discussions. Okay, so as we strongly ask you to use the chat when you want to make any comment or to put some questions to our participants. Right? So that said, I will then go through the first, so this opening from Professor José Fragata, and I will have now a small video to share with you.
Delivered immensely to our academia. I want...
Sorry, it was not in the beginning.
Good morning. Good morning. My name is José Fragata, and I'm the Vice Rector for Health at Nova University. It is a great pleasure for me to open this 4th International Conference from the NOVAhealth Chronic Disease and Infection Research Group. This group, among NOVA's most dynamic research groups, has been producing very high quality research and has delivered immensely to our academia. I want to compliment and acknowledge Professor Lígia Saraiva from ITQB and Professor Cláudia Conceição from IHMT, as all the researchers in the group, for their tremendous leadership at putting this group forward. Nova Saúde fosters collaborative research among Nova's organic units, and this particular chronic disease and infection group deals with diseases produced by non-vector infectious agents, which are becoming more and more prevalent. Chronic and infectious disease are indeed one of the major global human health problems that include neurodegeneration, inflammation, immunological dysregulation, and old and emerging microbial infectious diseases. Chronic diseases have been leveraged by the longer human lifespan, while globalization, migrations, exponentially increasing of travelers have been associated with an alarming escalade of microbes that are resistant to antibiotics. Even very recently, the Lancet, I think yesterday, or the day before yesterday, the Lancet came up with a paper where the emerging climatic changes are really changing the way this infectious diseases are spread globally, creating new threats. So this group promotes research, evidence-based knowledge and awareness to develop new effective and sustainable approaches and solutions to those same problems. The group fosters academic and social discussion in order to respond to daily challenges posed to patients as well as to the health systems and to their professionals. The topic for today's meeting is of a great equity, microbiota and health, as it is now known that our inside bacterial ecosystem does play a major role in our individual health, but not only and not less for public health. Truly, the multitude of interactions from mere digestion to the effect of antibiotics and vaccines all interfere with mechanisms of disease production that we now perceive in a more extensive detail. Today's meeting program includes relevant speakers, good people, and comprises the whole spectrum of microbiota, from induction mechanisms, psychiatric disorders, to food chain metabolism, oral poliomyelitis, vaccines, Legionella, and host cell targets to inflammatory bowel disease, also with relevant implications to public health. Last but not least, I wish, in the name of the university and my own, to acknowledge the cooperation with Pfizer, a dear collaboration that NOVA wishes to thank. It is indeed a good example of how the academia and industry can collaborate along projects and by sharing projects. Direct participations as today's with the talk by Pfizer on antimicrobial stewardship impacts on microbiota and health. This collaboration with Pfizer also materialized in relevant funding to research at NOVA in the field of chronic disease and infection needs and should be acknowledged and recognized. I wish to greet the organizers, the speakers, and the audience, and wish you all an enjoyable and profitable meeting. I need to apologize for not being in presence.
I think we got here some. Sorry for this.
As I would like, but some colliding events with participation of the other public university in Lisbon forced me to use this digital way to communicate with you, but this does not minorize my enthusiasm and even my gratitude to this brilliant group of researchers for putting up this extraordinary program. I wish you a very good meeting. Thank you very much.
So thank you to our esteemed Vice Rector. And I think now we can then start by our also esteemed first speaker, our keynote speaker, which is Professor Maria Augusta, and she's coming from Maria Augusta Coelho. She's coming from the Faculty of Medicine from the Porto University, and she will present us a nice talk about the prebiotics and the probiotics in psychiatric disorders, and also data, experimental data, and the clinical evidence. So I now give you the floor, Professor Maria Augusta.
Hello, everyone. Are you seeing my PowerPoint or no?
Not yet.
Not yet. I'm sorry. Oh, wait. Just let me get.
You are not presenting.
No.
No, it's getting. Yes.
Okay. And now it's there?
Yes.
Okay. Good morning, everyone. And first, I would like to thank you, the organization, and congratulations for your topic, a hot topic here in science and in medicine. And 2 words also for Professor Ligia and Professor Conceição, Cláudia Conceição, and Professor Ligia Saraiva, because they reached me some time ago, so I'm sure they must organize this meeting with very carefully. So thank you very much for your invitation, and I hope you enjoy the meeting. So they asked me to talk about. Is everyone? Are you listening? Okay. There is someone.
It was some interference. We will.
There is someone with an open sound. Can you please shut the sound? Okay. Paula, are you listening to me? Okay, the sound?
Yes, it's perfect.
Okay. So the idea is, I don't know if you, I am a pharmacologist, but I also am a psychiatrist. So I do like basic and the clinical work. And that's the way I'm going to see this, okay? I would like to speak about if there is any clinical evidence for to use prebiotics or probiotics in psychiatric disorders, okay? And which are indeed like your interest is a chronic disease in several cases. I have simple objectives. First, I will speak about simple and issues like microbiota and gut-brain crosstalking, and if we can find changes in microbiota in neuropsychiatric diseases. And the idea is to answer the question, is there a therapeutic effect for psychobiotics? And if yes, what is the strength of these results? So, simple ideas. First, I want to share with you one thought that I have. If one of you come to my consultation about, I don't know, 10 years ago, and you will tell me that you have live organisms in your body, inside your intestine, and that those organisms, live organisms, were interfere with your brain and with your emotions and with your behavior. I'm pretty sure that you could have could go away from my office with an antipsychotic drug. So you could have like a delusional idea. And now this is not a delusion idea. It's like, it's true. It passed the reality test. So it's not delusional. Yes, there is a collection of bacteria and virals and fungi that colonize different parts of our body, and we call microbiota, and this and also the complete genetic material that is encoded by microbiota, we call it microbiome. And this microbiome now is very important, because now we have different analysis capacity, namely these very important microbiome-wide association studies that in somehow have revolutionizing clinical investigations, and they provide a greater patient stratification and also new founding to find new biomarkers of diseases. It's like a new way of looking to diseases. And in fact, the microbiota, it gives these phrases that this idea that numbers do count, yes. And in fact, we have the numbers of microbiota cells outnumbers our human cells by approximately 1.3 to 1, and compromises a vast ecology like bacteria, yeasts, parasites, helminths, virus, and protozoaries. And in fact, they are very small, but the number are important here, of course. And so they exist. And another important characteristic is that this human microbiome is highly personalized. And that means we have different kind of profiles for microbiome, and each individual has also different species in different organs, like the oral cavity is different from the profile in skin or in colon, and so on. So this brings a problem for investigation, is that how can we take a representative sample of human microbiome? And this manuscript from Nature Medicine, it has already some years, but here in this graph on the left, you can see the microbiome state, and in the green, because the line in green is the evolution of microbiome state with years of age. But if we take a part of this line for a smaller one with a time days shorter, we can see a variation, and in the blue. And then if we take just a small time of like hours, there is also large variations. So taking a representative sample of human microbiome at any given site is challenging, because while microbiome is known to settle afterward, okay, but the composition can vary both over short term and long short term. So to take samples, the effect of rate of changes of varying species on ability to take a representative sample is these black lines. We have to take in account that microbiome can change over time also, and over hours. And today there is this idea that we can say it has very robust evidence that a variety of diseases are related or are implicated also. Microbiota is implicated in this disease, at least And we don't know if it is, sometimes we don't know what comes first, if it is the egg or the chicken, but examples of these included psychiatric, neurodegenerative, pain, distress, irritable bowel syndrome, stroke, addiction, and obesity. And a lot of diseases are, in fact, now can be looked at through a different window. Besides diseases, another important interest of microbiome is that we try to relate what happens with aging system, from birth through adulthood and aging, what happens with our microbiome and what happens in the brain. And in fact, it's the same kind of difference that happens, like we are losing variety, we are losing connections, both in the microbiota and both in the brain. Also with diseases, like I could say that most of the first diseases that were studied for microbiota or relations with microbiota were from metabolic disease, like diabetes, obesity, cardiovascular diseases. And in fact, we know now that in the intestine we can have different kinds of microbiota. And for instance, at birth, we have here, I'm not sure if you are seeing my pointer, but on the right side, we have a different dominant microbes at birth, adults or elderly people. Here in young babies, we have bifidobacteria and Streptococcus thermophilus. And then the dominant in adulthood are bacterioids, alistipids, bacterioids also. But this is changing to elderly. And in fact, during pathologic conditions, there is a dysbiosis of these bacteria that can lead to an increased state of inflammation. And there are also several ambiental causes for these increases in inflammatory stress in human body. And in fact, the profile of microbiota has been described as with changes, with important changes in several diseases. Most of them are in fact chronic here. Human gut microbiota are it's classically divided as two phyla, important phyla, Bacteroidetes and Firmicutes. But one thing that it worries me is that we can have species that are higher in some diseases, but those same species can come with lower levels in other diseases. So it's not easy to find one species for one disease. Perhaps the most one of important, interesting finding about this, it was like for Parkinson disease, the infection with Helicobacter pylori is very robust, the kind of evidence. And we have a lot of bacteria in our body, especially in intestine. Yes, it can be changed. It is different when we are ill, or perhaps it also contributes to becoming ill. But how come that microbiota can speak with the brain? How they communicate, the brain and the intestine and these bacteria? Now we know that it's not only what we eat or our diets. Of course, that would be the most important thing, but I am not sure. But a lot of things can change, can influence microbiota and can change brain. And the opposite is also true. our emotions can also influence these populations, like microbiome. If you all know that if we are stressed, if you have higher levels of anxiety, or if you have sleep less hours, you can also have abdominal symptoms. This is very typical, for instance, in children that have abdominal pains if they are with stress levels. You all know the idea of having butterflies inside your stomach. So there is a direct, in fact, a direct communication between the intestine and the brain. That is known for a long time, of course, but they can interact with each other, and it's well known that various behaviors know to be affected by microbiota, including cognitive, social behavior, stress, fear, and food intake. And this is related with two ways, okay? We know, for instance, that exercise can have beneficial effects on the brain, and we know now that microbiome also changes with exercise. For bad things, it's also true. I think you will talk here about, for instance, the use of antibiotics or other drugs. Like in psychiatry, now we know that a lot of psychiatric drugs, like antidepressives or antipsychotics, they change the microbiota profile. That's a reality. We don't know if it is good or bad, it depends. And there is a relationship between microbiota and brain, but how do they communicate? What is the language? What is the way they find this communication? In fact, microbiota, gut-brain signaling can occur via several pathways. And one important pathway is the immune system, because these bacteria are identified by our innate immunologic system. And they recruit the host's neurochemical signaling directly, also by enteric nervous system routes, and like the vagus nerve. You all know the experience with the vagus nerve and what happens if we get this nerve, and the production of bacterial metabolites. We also know about this, that some bacteria produce compounds that are essentially anti-inflammatory, like short-chain fatty acids. Other bacteria produce inflammatory ones that increase leakage between cells and provoke like an inflammatory state. So, and we have a lot of, I would say, actors in this movie, but in fact, all this knowledge help us to understand how bacteria can change central functions. Because you know that in the brain, we have a blood-brain barrier that it stops everything that can, attack neurons. But some of these small molecules, like short-chain fatty acids, can cross this barrier, and that's good. And the other compounds that have strong inflammatory effect can increase leakage in these barriers and facilitate the inflammatory state. A lot of investigators and groups are looking at this new way of thinking as a new therapeutic opportunity. And this is a very recent from Annual Review of Pharmacology and Toxicology, a very nice paper that I strongly recommend if you like this issue. And what we have in concrete, what we have about psychiatric diseases? Is there any evidence that microbiota is changing there? Oh, wait. Yes, we have a lot of evidence from animals, from experimental approach, basic, but also from studying microbiota in diseases, in psychiatric diseases. The most, I would say, idea that I think it's more logical to use is this about bacteria that produce this kind of compounds, fatty acids, like propionate and butyrate, because we know that if you have higher intake, higher levels of these fatty acids, they can activate directly receptors in the intestine, and they can increase an effect of anti-inflammatory effect and decrease in energy intake and decrease in blood glucose. And we know that when there is a disease that or a changing in the levels of these bacteria that produce these fatty acids, we have like a deviation of the response from the intestinal cells, and they have higher inflammation. We can have higher inflammation tone. We have an intake in energy, and we have also higher blood glucose. That's important for most of the chronic disease that do have like an inflammatory trait behind. And that comes not only diabetes, as you imagine, or cardiovascular disease, but also psychiatric disease, especially depression, okay? And that's why I bring this idea from also a very recent paper from Nutrients, where these authors explain how changing the microbiota to an anti-inflammatory profile with using probiotics, we can decrease neuroinflammation, we can increase some synthesis of neurotransmitters that can help with the humor, for instance. We can shut down or decrease the activity of hypophysis adrenal axis. You know, it's a very important response for stress, and we can decrease glucocorticoids like cortisol and the pro-inflammatory cytokines. So there is some evidence of this movie, of this image. It's not delusional, but sometimes we most of the results and experiments are being done in basic experimental, or in humans without disease. But they can have symptoms. For instance, you can have depressive symptoms, but you are not depressed. You can have anxiety symptoms, but you do not have a disease, you understand? And as a psychiatry, I thought that we should also know if this kind of prebiotic or probiotic compounds, if they also can be used for psychiatric disease or patients. For now, we know that changes in gut microbiota are observed in mood disorders, autism spectrum disorders, to a lesser, and that is most consistent. Not so consistent, but also found in obsessive compulsive disorders and schizophrenia. That is for psychiatric diseases. As an example, for instance, I bring here results from also this another Nutrients paper, where they found differences in the variability, in the diversity of species in patients with schizophrenia. One thing that I noticed, and I think you will notice if you study these issues, is that it's not always the same species that we found that it's changing. But I could say that most of the diseases studied, what we have is less diversity in the patients comparing to controls. Another strange finding is that if we transplant these bacteria, we call it fecal microbiota transplant from humans with schizophrenia into germ-free mice, so mice that are since birth they do not connect with the bacteria. And if we transplant fecal material from schizophrenia patients to germ-free mice, they cause a schizophrenic phenotype in animal. And this is also true for another kind of diseases. The most authors that describe this kind of experiments, they also found differences in signaling after the transplant in the central nervous system after transplant, like glutamate signaling. So changing the bacteria in the animals, we can give them also a phenotype of a brain disease, which is really a difficult concept to understand, I think. And okay, we have evidence that microbiota is different in several diseases, in several psychiatric diseases. But my question is there any evidence that if I try to change the microbiota in patients, do they get better? It's like, is there a therapeutic effect? And in fact, the term psychobiotics, it's coined by this group in 2013. It's a novel class of psychotropic as defined as live organism that, when ingested in adequate amounts, produces health benefit in patients suffering from psychiatric illness. And we have probiotics, symbiotics, and fermented foods, and that's the importance, for instance, of Mediterranean diet. So there is a lot of potential psychobiotic therapies. There are now a lot of new designation for psychobiotics, and I found this very important, nice one, is that psychobiotics are live biotherapeutics or substance whose beneficial effect on the brain are bacterially mediated. They are currently being investigated as direct or adjunctive therapies for psychiatric and neurodevelopment disorders, and possibly also for neurodegenerative disease. And they may emerge, attention, they may emerge as new therapeutic options in clinical management of brain disorders. So it's like a new way of looking to psychiatric disorders. I give you one example that I found interesting, this bacteria, Fecalium bacterium Prausnitzi. And it's interesting because this bacteria produces butyrate. It's another short chain fatty acid through fermentation of dietary fibers. As you know, butyrate, along with other fermentation-derived short chain fatty acids like acetate or propionate, they they have an anti-inflammatory effect, and then can be interesting for several chronic inflammatory diseases. For instance, in rats, also in humans, they can reduce cortisol levels and interleukin-6 level. So there is like a new interest in specific bacteria to promote human health. I as a psychiatrist, I want to know that if I can give to patients prebiotics or probiotics and they get better, okay? And so it's there a therapeutic effect of these compounds, of psychobiotics. And we have made a systematic review. And the first thing that we noticed that, because we were trying to see only randomized control trials, clinical trials in patients. And the patient should have a diagnosis in the classified as a psychiatric disorder. And the first thing we notice is that from thousands first and then 200 studies, most of the studies were for symptoms and were like not control, they were not randomized clinical trials. So we only found about 11 studies that were really, how can we say, strong enough to talk about a compound that changes symptoms in a disease. And but yes, we found that that's important. So most of the studies you will find are focused on persons that do not have a disease, but they have symptoms, which is different from to have a disease that can be classified and that we can, quantify the way you are ill or not. And I just have some tables. I brought tables from another review that they found similar results, but the table are more easy to read. So for major depressive disorders, you have here the control studies, clinical studies with probiotic treatment. And not another point is, what I find is that we don't have like a protocol for a probiotic treatment. Which agent, how many of them, how much should be the treatment? So you will find several differences. For instance, here they have used Lactobacillus acidophilus or L-casei, but then in the other they have used Helveticus. So it's not always the same. What does not that does not help. In depression, they have used almost always 2 weeks for 8 weeks. And yes, I can say that in psychiatry, the most robust effects are found for probiotic treatment are depression and anxiety. It's where we can talk about, right? Yes, I'm finished.
Thank you.
For other type of diseases like Alzheimer's diseases, that's where it's a new interest, since this is a neurodevelopment difference and there is a decline in our cognitive function. As you know, you will see some effects also, a multibiotic compound that for 12 weeks, it helps with some cognitions, but secondary outcomes are very important here in these kind of studies, especially because they can also help to change lipid profile and carbohydrate metabolism. But yes, we have some results. For schizophrenia, we have, neutral effects in terms of symptoms, but we have changes in secondary outcomes like Candida albicans antibody levels that these Candida is related, also in this area of knowledge, also related with higher frequency of symptoms, and it appears that probiotic treatment can help with changing the population of Candida albicans. And that's another kind of approach for autism spectrum disorders. As you know, it's an area where a lot of people put some interest in this bacteria. And but as you can see, it's very spoken, but we only have one important like clinical trial for autism severity. But yes, but with the patients with a psychiatric diagnosis, but with some important and interesting effects. So we can conclude at this point that also recent findings in psychiatric disorders are encouraging the use of prebiotics or probiotics in clinical practice still lacks sufficiently robust evidence. However, we can say that microbiome is definitely an actor in gut-brain interaction, and it's a fascinating new field in neuroscience. We would like to have more clinical trials to confirm therapeutic efficacy of psychobiotics. Thank you for your attention. I'm sorry to take more 5 minutes. Thank you.
Thank you very much. I really appreciated this talk. It was really new for me, but also very interesting. And I see from the audience, which also thought the same, we have already some questions. One is from Antonio Pedro Fonseca. So he's asking, since there is an epidemic use of anxiolytic and antidepressive drugs, should psychiatric and psychologists have their consultations with a nutritionist? A new controversial clinical approach?
Well, I think that we are in Portugal. Perhaps we would like to have at least a psychiatrist and psychologist. If we are talking a perfect world, and I believe it's possible, yes, it's very important to have a multidisciplinary approach. And here in psychiatry, I believe that the way people eat, the way people behave with food is essential to get better, because to eat is not just to ingest nutrients, of course. And so yes, please, I really would like to have more help from nutritionists. It's true, for instance, I work in a general hospital, and I have a nutritionist that also help us in psychiatric department. Essentially, she's worried about anoretic and bulimic patients. But yesterday we had a conversation about these prebiotics and probiotics, and she was not really like I almost felt that I was with a delusion idea. So not everyone knows this knowledge that we are talking about. Yes, please help me with that.
So the next question goes through this line of thoughts. Is from Ligia Saraiva. So Ligia is asking, how is the microbiota from different food habits influencing the evolution?
Well, now I think I'm not a specialist in that, but now we know that after when men start to be near water, you see with this Neolithic phase, started to eat more nutrients, especially these lipids that now we know they are very important, that they are present in fish, in shellfish. It's like when men became more close to the sea and to fish and to they evolve in terms of brain. That's most of the things I've talked about. I have a lesson, but yes, we can change our way of thinking according to what we eat.
Are you suggesting that those that live close to the sea, like us in Lisbon, are more clever?
Well, no, because if you if you can be there, but you could have bad habits of the way you have your food.
Yes, exactly.
But you know what I think is another important idea that I would like to leave with you is that medical doctors have this attitude that you should do that, you should not do that. And sometimes there is not so strong scientific data to do. So we should go more from prevention, like this idea, like having more diverse foods with colors, with vegetables, with olive oil. It appears to be a good choice for your health. Like giving the information and the person should choose, I think. There are some questions here. Like,
I'm reading them.
Okay.
But you are free to read them as well. It might be easier for you to read them. So Selena Madureira is asking which are the strains of bacteria that are elected to produce these medical compounds, and how do you execute a clinical trial since there is not a protocol? How is this adaptable to each patient? This is a nice question.
Yes, Selena, you make a lot of questions in the same idea, but it's very important what you have asked, because it's essentially what I'm afraid of is that this must be serious if you are talking about a therapeutic effect. So we must know which is the strain of bacteria, how much. It's exactly what I felt when I started to study this issue. I don't know. I think after like this bacteria that I show you, basic science can give you some choices. Like this bacteria works well in the rats. Okay, you should start with those that work well, and then we should start to do this with a scientific method. I cannot say at this point which bacteria and how, but that should be the way. You see, we should be serious about the way we are investigating these compounds. For patients, I don't think it's different, because they get much more complicated compounds. So to it, I saw a very interesting study to finish. They gave patients with mania. I don't know if you imagine what is a patient with mania, but it's someone that is completely out of good mental health. It's very difficult to deal with them. And they found that if they give a special strain of bacteria and the other control, they give a similar liquid.
And how did they give the bacteria?
Ingested with like a yogurt, you see?
Okay. Mm-hmm.
And I don't imagine how they took them because they are always very suspicious. Anyway, but they could, the control, comparing the patients with the control also with mania, the patients who took this, I will say probiotic, mm-hmm. had less 3 days of internal hospital. They could go out sooner. I thought it was unbelievable as a psychiatrist. Mm-hmm. And when I talked to my colleagues, they say, it's not true, Augusta. Otherwise, Hospital São João will give you a lot of money to have this compound. So you see, we are still doubting about these ideas. That's what I feel. So they must become serious. How to do these studies, I think.
Thank you. So a question for myself, very basic. Psychobiotics, you gave the definition, but when they are given to the patient, are they the bacteria themselves or extracts from the bacteria?
So as in the first time we use the term psychobiotics, should be bacteria. But now you can use the term psychobiotics for a lot of type of like fibers, like kinds of nutrients. Everything should go finally from bacteria, but it can be also prebiotics, for instance. Okay.
Okay. So thank you very much. So I invite all our colleagues here in this conference to, if they have more questions for Professor Maria Augusta, to use the chat or use the Telegram, which is another chat that we have that is available through our website to continue this conversation. So thank you very much. And it was a pleasure to listen to you and to all this and learn with you. Thank you, Professor Maria Augusta.
Thank you, It was very nice for me also.
And now I think it's my colleague Maria Gracia. No, sorry.
Hello, everyone. In fact, it's me, Andrea Light. Just to briefly introduce myself, I'm an assistant professor at the National School of Public Health, and I'll be moderating the next two blocks of presentations. Following this exciting presentation from our keynote speaker, we now move to our flash presentations. So we've selected presentations from each one of the NOVA University units, and you'll see the breadth of research we have in our university, ranging from lab-based research to observational research in various areas. So we now move on to the first flash presentation from Mariana Camões from ITKB. I remind our presenters that they have 10 minutes. I'll kindly ask you to stick to your time so we can stick to our schedule. Mariana is now sharing her screen. Thank you very much for your time, and we'll have now the talk following dissemination of microbial populations through the food production chain. Just before starting, I remind our audience that you can use the chat to put your questions, and following this, each two presentations will have some discussion time. Thank you very much, Mariana, the floor is yours.
Good morning. Do you see perfectly my presentation in your screen? Yes. Okay. So my talk is entitled Following Dissemination of Microbial Populations Through the Food Production Chain. As you know, when you get sick, you get antibiotics and you can develop resistant bacteria in your gut. And the same way happens to the animals. When they get sick, they can get antibiotics and obviously can develop resistant bacteria in their guts. And what happens is that the drug-resistant bacteria that can remain on the meat from animals were not handled or cooked properly. The bacteria can spread to humans. And is this precisely step that was the focus of our project called Farm to Fork that I'm going to talk about today in this presentation. So this project, Farm to Fork, includes 5 resistant bacteria that are named by the World Health Organization priority pathogens list for the research and development of new antibiotics. including carbapenem-resistant Acinetobacter species, carbapenem-resistant Pseudomonas species, carbapenem-resistant Enterobacteriaceae, and also ESBL producing, and also vancomycin-resistant Enterococcus species, and methicillin-resistant Staphylococcus aureus, named MRSA. So this project, the research have some research questions and goals, and some of these research questions are going to be exposed here in this presentation. And the first one is how does the microbiota change over the food production chain? And at which steps are humans exposed to higher loads of antimicrobial resistant bacteria? And can whole genome sequencing, metagenomics, and culturomics help to detect new sources and routes of dissemination? And the goal of this presentation is also to describe the bacterial community structures at the different sites along the pig meat production process, using a combination of traditional culturing techniques and the 16S RNA gene sequencing. So to achieve these goals, we performed the sampling in 4 days. On day 1, we went to the slaughterhouse and we performed a sample from pig ear and pig rectum. And at day number 2, we obviously went to the slaughterhouse and sampled meats, surfaces, dirty, clean hands of workers that are working on the splitting zone. And on day 3, we went to the butcher and we collected some sticks of pork shoulder, and we delivered these sticks to the consumers. And on the day 4, we gathered the cooked samples from the cooked sticks, dirty, clean hands of consumers, and the ends after the meat seasoning. So after the sampling, we performed the culturomics approach. That basically this performed the serial dilutions, and we played these dilutions on selective media and selective media plus antibiotic for these 5 resistant bacteria. And after that, we determined the CFUs, and we pick up to 10 colonies, and we determine the phenotype based on to determine the pathogenicity and the resistance of these resistant bacteria. In parallel, we also performed the genomic analysis by whole genome sequencing in order to analyze the transmission between these resistant bacteria. Obviously, in parallel, also we froze the suspensions of these samples at -80, and after that we performed the DNA extraction in order to perform the metagenomics experiments. We included 110 samples for the metagenomics experiments. And I forgot to say that we included 185 samples for the culturomics experiments. In the metagenomics experiments, we analyzed the amplicon, the B4 region of the 16S RNA gene. We analyzed the data using the pipeline CHIME2 that allow us to characterize the bacterial community and calculate the diversity and the relative abundance of our samples. Also, we will go to perform the shotgun metagenomic to determine the load of the resistant genes, but these data are not shown in this presentation. So I decided to divide my results in 2 main parts. The first one is the diversity and the bacterial loads. The 16S sequencing, this is the results of the analysis of the phase phylogenetic diversity that we obtained after the 16S analysis. And after analyzing this box plot graph, we conclude that amplicon sequence variance richness was significantly different among sample types, and the highest richness was for the pig samples. In this graph, we show the relative frequency, the relative abundance of all the samples that we studied. And we have a high bacterial frequency load of in the pig samples, and as you can see here by the colors and by the diversity of like the different colors, the diversity of colors, that means that diversity of species and then the taxonomic different taxonomic classes, and was the most diverse was the pig samples. And I would like to show you also the relative frequency to the different resistant bacteria that we characterized by culturomic approach. For example, the Staphylococci were present throughout the pig processing chain, as well the Acinetobacter species, as well the Enterobacteriaceae, were present throughout the pig processing chain, as you can see, and also the Enterococcus species. Pseudomonas species were almost absent in pig samples. We are only found in some pig samples, but a few amount, but it's present in the remaining sample types. So after, now I'm going to show you the results of the Culturomics approach. After the Culturomics approach, we observed that a high load of putative antimicrobial resistant bacteria were found along the pig processing chain. As you can see here, there are not not antimicrobial resistant bacteria in sticks, cookies, and pig rectum. When we analyzed by different genus that our resistant bacteria that we detect on the culturomic approach, here we can show the different bacteria resistant that we studied, and we observed that the Carbocation resistant Pseudomonas species and the vancomycin resistant Enterococcus species were present in the majority of the samples. If you can see deeply this information, you can see that the Enterobacteriaceae, KPC or SPL were present mostly in surfaces, pork cuts, and consumers. The MRSA were present mostly in pigs, humans, and pork cuts. The Carbocation resistant Acinetobacter species were present in the majority of the sample types, as well the vancomycin resistant Enterococcus species, and as well the Carbocation resistant Pseudomonas species. Now the second and the last part of the results to show you the bacteria transmission. So I'm going to show you results. If you remember, we did also the genomic analysis by the whole genome sequencing to see the transmission. We decided to perform the transmission analysis only in the most prevalent strain, the most prevalent resistant bacteria. And in this case, we detected that was the Carbocation resistant Pseudomonas species and the vancomycin resistant Enterococcus species. So first, I'm going to show you the Carbocation resistant Pseudomonas species results. Sorry. Okay. So you can. These results are results from our master student that we have in our lab, and Abdallah. She included 26 isolates and created this heat map. The blue colors reveal that are very related isolates, and the red ones are not so related. And she defined the transmission cutoff less or equal than 14 SNPs to classify a transmission event. And she concluded that 43% of the samples involved in cross-transmission, and the transmission occurs within different pigs and pork cuts, and between pork cuts and the raw meats. As I said before, we also analyzed the cross-transmission in the vancomycin-resistant Enterococci species, and we included in this analysis 58 isolates. We also create a heat map. We see these blue colors are the most related isolates. The red one are not so related. And we create an interaction networks with 16 samples very related. And the cutoff that we applied was less than 74 SNPs. And we concluded that out of 31 cross-transmission events, 70 are human cross-transmission events. So Pseudomonas, Carbocation resistant Pseudomonas, and vancomycin Enterococcus species, resistant Enterococcus species are the cross-transmission was very frequent in these two resistant bacteria. Regarding the results that we obtained with the 16S sequencing about the beta diversity, that means that we are studying now the bacterial community structure of our samples, and are represented here in these plots. The consumers are in the red dots, pigs in the yellow dots, the workers are in the purple dots, meat in blue dots, and surfaces on the green dots. And if you make some zoom, you can see that the workers that are the purple dots cluster with different sample types, here with pigs, the surfaces, and meat, and with the consumers. So it means that the human are exposed to antimicrobial resistant bacteria throughout the food processing chain.
Mariana, please,
it's the last
running out of time.
I'm sorry. So the last result that I'm going to show you is the similarity between clean and dirty hands, to see if the hand washing is an efficient procedure to clean. And indeed, we make a zoom to this graph to explain better that it's the workers, dirty hands and clean hands are very similar. The diversity of the microbiota that we found is very similar, and also the consumers have the same results. So the hands washing was not efficient. So the take-home message is the high load of antimicrobial resistant bacteria was found along the pig processing chain. The carbapenem resistant Pseudomonas species and vancomycin resistant Enterococcus species were detected throughout the pork production chain. Pigs was the sample type that showed the highest genetic diversity. Antimicrobial resistant bacteria transmission occurs between humans and different steps of the pork production chain. Hand washing was not enough to decrease the antimicrobial resistant bacteria load and modify the species diversity. And finally, pigs have a unique microbiome pattern and appears to be the source of antimicrobial resistant bacteria to the other steps of the pig production chain, including humans. And I would like to thank to my colleagues of the Bacterial Evolution and Molecular Epidemiology Lab, and also members of the Faculty of Veterinary Medicine and Copenhagen University Hospital in Iverdova. And thank you. Sorry for the time.
Thank you very much, Mariana. I remind you that you can ask any questions you might have to our chat. We will now move to the next presentation, but we'll have some discussion time after that. I'll now introduce João Araújo from Nova Medical School, who will present lipids, intestine, and gut microbiota interactions, ménage à trois. Thank you very much.
Good morning, everyone. First, I would like to thank the organizing committee for having invited me, especially to Claudia, which was the interface between me and the meeting organizers. So my presentation is about lipids, intestine, gut microbiota interactions, and ménage à trois. So the intestinal digestion, absorption, and metabolism of lipids is quite a complex process. However, I can briefly summarize you with this picture on the left side of the slide. So first, lipids from the diet are broken down into small vesicles called micelles, and this process is performed by bile acids. So bile acids transforms this big fat lipid droplets into small lipid droplets called micelles. Then at the same time, triglycerides, which are the main lipids from the diet, are hydrolyzed into fatty acids and monoacylglycerols. Then these fatty acids and monoacylglycerols will be taken up by the enterocyte, the main cell from the intestinal epithelium involved in the absorption and metabolism of lipids from the diet. And once inside the enterocyte, fatty acids and monoacylglycerols can be either oxidized or travel to the endoplasmic reticulum, where they will be reassembled again in triacylglycerols. Then triacylglycerols can have one of two destinations. Either they are stored as lipid droplets in the enterocyte, or travel to Golgi to be fused with other lipoproteins and will generate intestinal lipoprotein, either they travel to the Golgi to fuse with proteins to generate lipoproteins, which at intestinal level are called chylomicrons, and then these chylomicrons can be secreted to the bloodstream via the lymphatic system. Okay. So perhaps the first study the first high quality study that consistently demonstrated the connection between this kind of intestinal lipid metabolism and gut microbiota was this one from 2018. In this study, the authors showed that germ-free mice, so mice without a gut microbiota, okay, either they ingested a high-fat or a low-fat diet, okay, they showed lower circulating levels of triglycerides and cholesterol, and higher levels of fecal triglycerides and cholesterol. So this study showed that the gut microbiota is quite important for the efficient digestion and absorption of lipids, okay? And in fact, the gut microbiota seems to modulate the intestinal lipid metabolism by two ways, I would say. First, the gut microbiota interacts with the absorption and secretion of lipids at the enterocyte level, and also can modulate bile acids metabolism. And I will give you two simple examples of these two kinds of interactions. So if you look at the top, you see in blue enterocytes. In these two studies, the authors exposed enterocytes here in blue to controlled conditions and to two strains of commensal bacteria, Lactobacillus paracasiae or E. coli. And as you can see here, after exposing enterocytes to these two strains, they exposed cells to this to a lipid probe, okay, to a fatty acid lipid probe that you can see here in green. And what you see is quite striking. So when you expose enterocytes to Lactobacillus paracasiae, you can see an accumulation of lipid droplets here in green. So we have more lipid droplets and bigger lipid droplets. Also, the lipid secretion of the lipid secretion of chylomicrons is also decreased. When you expose E. coli treated cells to lipids, the behavior is totally different. You can see that when enterocytes exposed to E. coli, there was almost no lipid droplets observed. So the size and the number of lipid droplets was quite smaller compared to Lactobacillus paracasiae and compared to controlled conditions. And then the authors tried to uncover the effectors responsible for these effects of Lactobacillus paracasiae and E. coli. And what they discovered was that Lactobacillus paracasiae, as Augusta said in the very beginning, Lactobacillus paracasiae was releasing a short-chain fatty acids called lactate. And this lactate was able to block fatty acid oxidation in the enterocyte. In E. coli, on the other side, was producing acetate. And this acetate was stimulating fatty acid oxidation by stimulating this very important pathway, AMPK, PGC1 alpha, and PPAR alpha pathway. Regarding the bile acids, so this is still not very well understood, but a very recent study using germ-free and conventionalized mice show that microbiota, the fact that microbiota is able to metabolize, in this case to deconjugate bile acids, can inhibit the synthesis of primary bile acids in the liver. And how does this process? It's a bit complicated, but I will try to explain to you. So basically, the microbiota is able to deconjugate primary bile acids, and these deconjugated bile acids at the intestinal level can stimulate farnesoid X receptor signaling, which can then increase the expression of this fibroblast growth factor, which by traveling to the portal vein to the liver can activate a series of intracellular signaling pathways that will culminate in the inhibition of cholesterol hydroxylase, which is the first step in the synthesis of primary bile acids from cholesterol. The authors were not able to identify the microorganisms responsible for this effect because they only saw this effect comparing germ-free versus conventional mice. However, one strong hypothesis is the Bifidobacterium and Lactobacillus species, because these species have bile salt hydrolysis, an enzyme that is able to deconjugate the bile acids. So the main take-home message of these two recent studies was that, in fact, gut microbiota may impact several steps of intestinal lipid metabolism, and then this can have quite important implications in the development of bacterial and metabolite-based therapeutic interventions against a wide range of metabolic diseases, ranging from obesity to even malnutrition. However, more data is still needed regarding these two aspects. So we need to understand more the impact of gut microbiota on other stages of intestinal lipid metabolism, especially stages at Golgi and endoplasmic reticulum in the enterocyte. And we also need to understand if the gut microbiota influences the lipid metabolism, either if we are talking of saturated fats, unsaturated fats, for instance, polyunsaturated fats, or trans fatty acids. And that's it. Thanks for your attention.
Thank you very much, João. I remind our audience that they can ask some questions to our presenters. We'll have some time now for discussion while our participants think and digest this information. There's a lot of food for thought. I have myself one question for each one of our participants. So, Mariana, regarding your conclusions, what clues for action do we have from your conclusion? So how can we apply these results in practice? Can we somehow have some intervention in food processing chains, in pigs in particular, to try to prevent this source of antimicrobial resistance, for example? And how far are we from having such an intervention implemented?
Okay, so indeed, if you cook it well, for example, the meat, and manipulate it properly, the meat, it's true that you don't have any health problem, because if you cook it the pork meat, all the resistant bacteria died, at least. The other one, only we can say that the only families or the only consumers that have problems and have some high load few of bacteria in the samples was because they don't manipulate properly the meat. So it's not indeed a big concern at now, at least. But for example, if you have in the food processing chain, you observe in this case that our objective is to compare the strains that we obtain in this food processing chain in each step, and compare also to the clinical isolates that I don't present here the results, for example, and to see if they are similar or not similar, and if they share some resistant determinants and virulent determinants. And maybe if you see the graph of the workers and see that they transmit the resistant bacteria throughout the food processing chain. It's worrisome because of that, because if you don't manipulate the meat properly, you could have a health a problem in your hands, because you are manipulating antimicrobial resistant bacteria in the meat. So you have to wash properly your hands, because some hands when are also well clean, you can also remove the bacterial diversity of your hands. So it's a good way to avoid contamination, and also to cook properly the meat. You can avoid this contamination. However, if you don't have these two situations controlled, you can be colonized or ingest the resistant bacteria. It's a problem by that.
We also have, thank you, Mariana. We also have some questions here from Ligia Saraiva, who's asking if some antibiotics are not degraded by heat, so they can also pass to us as well. And if washing hands does not influence the results, which should be our messages.
So some antibiotics are not degraded by heat, it's true, but indeed we have to study another thing apart from the antibiotics. It's that the cell viability when you eat the meat. Maybe with the metagenomics study we conclude that, because you have some bacteria that are there, and in the culturomics you didn't observe. So what happens? You cook the meat, you supposedly kill the bacteria, the resistant bacteria that have the antibiotic, but you can see that it's present in the metagenomic analysis. Maybe because the bacteria died, but the DNA is there, and the antibiotics obviously also are there, the antibiotics resistant determinants. So it could be a problem, yes, because as Ligia said, that are not degraded by heat. Even the bacteria are degraded, but are there and lyses the entire cell content. So they are available in our body, no? And if washing hands does not result, which are the advices. Indeed, I forgot to say in the presentation that we have some results in our lab that that we found the resistant determinants for biocides and for sanitizers, like efflux pumps and another mechanism that are try to make difficult the washing hands be efficient. But we have some cases in the food chain process that if you that they wash it properly the hands, and the diversity is totally different from what we expected. So and decrease the loads. So maybe if you wash the hands properly and with but obviously we don't eliminate completely what you have in your hands, because as I said before, even these bacteria could have some resistance to biocides or to sanitizers that you can use in our homes or even in the slaughterhouse. So we have to discover new sanitizers or new ways to new biocides to come to treat this problem, because it's difficult, because they are resistant even to the biocides and the sanitizers. So it's not easy to eliminate, but to decrease is possible.
Thank you. Still some unaddressed and unanswered questions that certainly we will continue to follow. Thank you, Mariana. One question also to João. You present very interesting results, and you mentioned some possible development of therapeutic interventions against obesity, atherosclerosis, and malnutrition. how far are we from this development? Are we still at very early stages? What's your thoughts?
Well, I'm not sure if I have all the knowledge to answer your question, but I think we are still, based on my conclusions, we are still in the early phase of developing this kind of products, because still we need to understand which steps of lipid metabolism are influenced by this kind of bacteria, and we also need to identify perfectly the effectors. Is it only lactate and acetate? Can we do a formulation in which we do not give the bacteria but only the effector, or perhaps both? So we are still in an early phase trying to identify all the targets of lipid metabolism, and then also the bacterial strains and the effectors.
Thank you very much. We have no further questions in our chat, but we can continue our discussions through Telegram. So I ask you all to join through the link available in our website, and we can continue our network also with the poster presenters later on. We now move to the next group of flash presentations, so more focused on issues with respiratory microbiomes. I'll present now Marcia Medeiros from the Institute of Hygiene and Tropical Medicine, who will present oral poliomyelitis vaccine boost drives changes on gut and respiratory microbiomes of infants. Thank you very much, Marcia. The floor is yours.
Good morning. I will share my screen. Okay, good morning. It's a pleasure for me to be present here. The results of the micro-OPV study, an observational cohort study showing that OPV is associated with changes in gut and upper respiratory microbiomes of infants. Among the several factors that have impacted on child mortality rate since the beginning of the last century is undeniable the role of vaccines against preventable infectious diseases, as we can observe here with the decrease in child mortality in children under age 5 due to tetanus, pertussis, or measles. Additionally, researchers have found that live attenuated vaccines, specifically measles vaccine, BCG, and oral polio vaccine, may protect against non-target diseases, conferring an extra protection implicated in non-specific reductions in child mortality. First evidence came from measles vaccine in randomized clinical trials and observational studies in 80-90 decades in Guinea-Bissau. Potential mechanisms of BCG non-specific effects has been described based on experimental studies in human and mice, showing that BCG is able to induce innate immune memory, the so-called trained immunity, and an heterologous adaptive Th1 and Th17 adaptive responses due to vaccination. And oral polio vaccine has been associated with decreases in the bacterial and diarrhea for bacteria, and antimicrobial peptides have been found in the stool of newborns after the first OPV dose at birth. To explore in more details the non-specific effects of oral polio vaccine, a large cluster randomized clinical trial has been conducted, the RECAMP OPV, involving with 10,000 infants from 0 to 8 months by researchers of the Bandin Health Project and University of Southern Denmark in Guinea-Bissau. And to answer a major question, if non-specific effects of oral polio vaccine may be driven by changes in gut or upper respiratory microbiomes of infants, a collaborative research was established with researchers from the Institute of Hygiene and Tropical Medicine and Statens Serum Institute in Denmark. The micro-OPV study, as a sub-study of the RECAMP OPV, comprised a field work that was conducted in Guinea-Bissau from August to December in 2018. Infants from 4 to 6 months enrolled in the RECAMP OPV and presenting no acute infectious disease was recruited over 3 regions of the Guinea-Bissau, Oyo, Cacheu, and Biombo. Micro-OPV study comprises 2 steps. This first field work, the enrollment and follow-up, distant 2 months late, 2 months distant. In the enrollment, infants from intervention clusters and infants from control clusters were recruited. Epidemiological and clinical data were collected, followed by nasopharyngeal and fecal content. And infants from intervention clusters received an OPV dose, whereas infants from control clusters received no intervention. 2 months later, these infants were analyzed again, epidemiological and clinical data were collected, and another set of biological samples. And 47 infants in each group completed these 2 steps of the field work. And the subsequent analysis were performed with this set of 94 infants. Laboratorial processing of the biological samples included obviously extraction of DNA and an analysis for deep sequencing analysis of the V3 and V4 regions of the 18S ribosomal RNA gene. After a preprocessing of the sequencing data, a number of amplicon sequencing variants were obtained, and they were characterized according alpha diversity and beta diversity parameters and statistical tests, and the reminiscents of the DNA were deposited in our biobank biotropical resources. Regarding the participant profiles of the infants in 2 groups, intervention and control, we didn't find any statistical significant difference. And it's important to observe here the gestational age, the delivery mode, and the feeding history that also determinant for the colonization process and the microbiota composition that we will see. And regarding the alpha diversity, we choose 2 indexes for characterizing this alpha diversity, the Shannon and the inverse of Simpson index, and we can observe an increase in alpha diversity in both groups over time without any difference regarding the magnitude of changes in both groups for gut microbiota, and regarding the respiratory microbiota, a significant increase in alpha diversity through Shannon index just in the intervention group. But when we compare the magnitude of changes over time, we didn't find any significant difference. So this means that first, alpha diversity is increasing, and it's corroborating the colonization process of these weaning infants recently introduced to more solid foods through porridges. But when we analyze the microbiota composition in gut and respiratory microbiota regarding taxa with relative abundance greater than 1% that significantly changed over time, we can see some differences in the intervention group and the control group. Both groups presented a decrease in the main metabolizers of the human milk oligosaccharides, the Bifidobacterium longum. And this was equal in both groups. And an increase in the Bacteroidetes and Prevotellacea family in both groups, but with a slightly different way. Here we observe a more balanced equilibrium before these two most abundant phyla and organisms belonging here. And regarding the Prevotellacea family, we can observe in this group a more diverse number of genera belonging to this family that increased over time, different from here that we can observe just one genera that increased. And another important difference was the decrease in some organisms that are considered more pathogenic or more opportunistic in the group that received vaccine, different from here when we observe an increase in some pathogenic and more opportunistic and some species that we are expected to occur in more older stages of life. Regarding the differences in respiratory microbiota, we can observe in the group that received vaccine an increase in the most dominant taxon expected to occur in these infants under this age, and the reduction of some opportunistic and more pathogenic organisms and species. Besides this same decrease observed in the control group, we observed here again an increase in more opportunistic and pathogenic bacteria from two important families causing respiratory infection in infants, Streptococcus and Haemophilus. And also, this is expected to be occurred in more older stages of life. So we can conclude with this micro-OPV study that OPV revaccination versus no revaccination is associated with changes in gut and upper respiratory microbiomes that suggest a potential OPV beneficial effect, enhancing and favoring the colonization for most abundant commensals involved in important metabolizers and production of short-chain fatty acids, for example, important for the development of the immune system and maturation of the immune system, as well impacting in the gut-brain axis. Also, we need a sequencing methodology allowing species discrimination to validate OPV against colonization for more pathogenic and opportunistic groups. Also, it will be very informative, repeatedly evaluations of nasopharyngeal and fecal contents to follow this OPV modulation of the colonization process and develop of the immune system. And it's important to improve vaccination coverage in these infants. I would like to acknowledge every people directly or indirectly that participated in this study, and also the participants in Guinea-Bissau. And thank you very much for your attention.
Thank you, Marcia. We will now move on to Irina Franco from Faculty of Sciences and Technology. She'll present us Lutonella Pneumophila and its host cell targets, what's the microbiome got to do with it. Thank you very much, Irina. And I remind our participants that they can also ask questions to both Marcia and Irina. Irina, the floor is yours.
Thank you. Okay. Okay, so can you hear me? Yes. Okay, first of all, I would like to thank the organizers for the invitation to present my work here today. And I'll be talking about Lutonella Pneumophila and its targets in host cells and how the surrounding microbiome shaped this interaction. So, okay. Okay, so Lutonella Pneumophila is a gram-negative, rod-shaped bacterium. It's an environmental pathogen. It's found in aquatic environments where it parasitizes its natural hosts, which are many different types of amoebae. But when people inhale contaminated aerosols, Legionella is also able to invade and replicate inside human lung macrophages, causing a severe type of pneumonia known as Legionnaire's disease. And the sources of these contaminated aerosols are essentially man-made, moist and warm environments, such as spas, showers, or cooling towers from factories, or air conditioning systems. And Legionella pneumophila is a facultative intracellular pathogen that is able to invade host cells and replicate in a specialized compartment called the Legionella containing vacuole, or LCV. And the formation of this remodeled compartment is absolutely essential for the bacteria to avoid killing by the host cell. So immediately after uptake, the Legionella containing vacuole, or the LCV, avoids fusion with the endocytic machinery and later with lysosomes. And the LCV is able to recruit vesicles that traffic between the ER and the Golgi, and it also recruits mitochondria. And in this remodeled niche, this remodeled phagosome, the bacteria are able to multiply until they escape the host cell and they start new rounds of infection in neighboring cells. And the major virulence factor of Legionella is a type 4 secretion system, which is required to inject bacterial effector proteins directly from the bacterial cytosol into the cytosol of the host cell. And in Legionella, there are over 300 of these proteins, and their function is to hijack functions in the host cell, such as cytoskeleton dynamics, vesicle trafficking, transcription, translation, and many other pathways. And in Legionella, a particular feature of these effectors is that many of them are similar, very similar to eukaryotic proteins or contain the so-called eukaryotic-like domains. So how did Legionella pneumophila acquire all these eukaryotic-like proteins? So it has been along millions of years of coevolution with the environmental microbiota. So during infection of amoebae in the environment, Legionella is able to acquire genetic material from the hosts, but also from other co-infecting bacteria and even from co-infecting viruses. So the acquisition of this foreign genetic material led to the existence of these effector proteins, which have eukaryotic-like domains, and these proteins are able to mimic eukaryotic protein functions. So this ultimately led to a fine-tuning of the interaction between Legionella pneumophila and its different hosts, allowing the replication of these bacteria in many different hosts by using different sets of effectors depending on the hosts. And this is also the reason why Legionella pneumophila has become an accidental human pathogen. So Legionella pneumophila is perhaps mostly known for causing very large outbreaks. And actually the second largest outbreak ever took place in Villafranca, Chile, a few years ago, you may remember this. And it caused the first reported case of a person-to-person transmission. And this outbreak was caused by a novel Legionella pneumophila strain, the strain PT-VFX, which was sequenced, and it shows a new genetic organization. So we wondered if this new Portuguese outbreak strain with this new genetic backbone could also have new virulence mechanisms. And we are particularly interested in identifying and characterizing new type 4 secretion system effectors. And to do this, we initially asked the group of João Paulo Gomes at the Instituto Ricardo Jorge to look in the genome of this strain and try to find candidate genes that would encode previously unknown effector proteins. And they found several candidates, and we tested them to see if they were indeed effectors translocated by the type 4 secretion system. And I don't have time to go into detail about this translocation assay, but I'll just say that we found two new effector proteins in Lutonella Pneumophila. This protein, VFX05045, and another one, protein VFX145. And I'll be focusing on this last one for the remainder of this talk. So a search in the genomes of other strains from Lutonella Pneumophila revealed the existence of homologs of VFX145. So it seems that this effector is not exclusive of this outbreak strain. When we compared the amino acid identity, we found that this particular protein, LPP3070, which is encoded by the Lutonella Pneumophila strain Paris, is the one showing the highest degree of identity at the protein level. So they are about 94% identical. And we tested this protein to see if it was also an effector protein using the same translocation assays. And we confirmed that this protein is also an effector. So to try to understand the function of these two proteins during infection of host cells, we started by assessing where they were localizing in eukaryotic cells. And we did this by expressing protein fusions, either of the VFX protein or the homolog LPP protein to GFP, and expressing these fusions in mammalian CHO cells. And we found that the VFX protein is very clearly localized in the nucleus of eukaryotic cells. If you see here, it overlaps perfectly with the DAPI stain that stands for DNA. But we were very surprised to see that its homolog, which is an almost identical protein, does not show this localization. So it's not localized in the nucleus, it's spread in the cytosol of eukaryotic cells. And to confirm that these two proteins show these different localizations during infection of macrophages, we again expressed them, this time fused to an HA tag, and used the corresponding Lutonella strains to infect macrophages. And these are the images I'm showing here. So we confirm that the VFX protein from the Portuguese outbreak strain, again, it very clearly localizes in the nucleus of macrophages. But again, we also confirmed that its homolog from the Paris strain does not localize in the nucleus. It spreads throughout the cytosol of macrophages. So we were wondering how such similar proteins could have such different localizations in the host cells. And to try to understand which regions of the proteins or which amino acids made VFX go to the nucleus and LPP remain in the cytosol, we expressed many different versions of these two proteins, either with single amino acid substitutions or encoding only particular regions of the protein, or we also constructed chimeras containing one part of the protein and one part of the other. And we expressed all these constructs as GFP fusions, and we measured the ability of all these proteins to still localize in the nucleus, and which is shown here in this graph. And to cut a very long story short, what we found was that there was a particular region in the C-terminal of the VFX protein, so localized here, that was absolutely essential for this protein to be transported to the nucleus. And when we compared the amino acid sequence in this region of the VFX protein with the one in the LPP protein, we found that there were several amino acids that were different. So we took the wild type VFX protein and we mutated these amino acids one by one, and we were able to pinpoint 2 amino acids that are essential for the nuclear transport. And these are this arginine here and this isoleucine here. So if we change any of these 2 amino acids, the protein, instead of being transported to the nucleus, it remains in the cytosol of eukaryotic cells. And very interestingly also, this region here does not contain any known nuclear localization signal. So it may constitute a new motive for proteins to be transported to the nucleus of eukaryotic cells. So just to summarize this work, we identified a new Lutonella Pneumophila effector, the protein VFX145, which is targeted to the nucleus during infection of macrophages, but a homologue from a different strain, the protein LPP3070, does not show this localization, so it remains in the cytosol. We were also able to pinpoint 2 amino acid residues, an arginine and an isoleucine, that are key to this nucleotropism of the protein. And on a more broader view, this work supports the idea that in different strains of Lutonella pneumophila, homologous and highly identical effector proteins still may play different roles during infection. And although effectors may originate from the acquisition of genetic material from the surrounding microbiome, along evolution, they can be fine-tuned to adapt their function to different hosts. So finally, I'd just like to acknowledge Jaime Motas, Infection Biology Lab here at FCT NOVA. Inês and Sofia did part of the work I've shown here today. Also thank our collaborators from the Instituto Ricardo Jorge and funding, and I thank you all for your attention, and I'll be happy to answer any questions.
Thank you very much, Irina. We'll now move to the discussion, and we have several questions from our participants, starting with some questions to Marcia. Ligia Saraiva is asking, you mentioned increase on antimicrobials, where do they come from? And also if polio vaccine positively impacts on gut and respiratory microbiota, which one is more influenced?
Okay, thank you. This study that identified antimicrobial peptides in the stool of newborns after the first dose is the human L-alpha-acetylcysteine 37. There is an antimicrobial peptide produced by the human gut, and there are several actions, and it's important that it avoids the colonization and forming of biofilms in the gut. And this is mainly for these bacteria such as Escherichia coli or Shigella that needs this interaction with the mucosal cells, and it's important to avoid this process. And regarding what the most was impacted with the oral polio vaccine, if gut or respiratory microbiota, so based on our results, I can answer that both microbiota were impacted. In fact, the most that presented, for example, this increase in commensal bacteria, that it's important for short chain fatty acids. We can we observe this in the gut, but in both microbiota there was a decrease in important groups that are considered more pathogenic and opportunistic. So my answer is that both were affected, positively affected. And the other questions, what is the, it's important, one thing that it's important to mention that is, for example, oral polio vaccine is planned to be removed from the routine immunization due to some rare cases of paralytic polio due to vaccinal virus. And our results show that it's important to maintain this vaccine in campaigns, that because these changes in both microbiotas can over time influence the response and the decrease in some infections or the chances of survival for these these affected children. I don't know if I answered all of them.
I believe you were addressing Claudia's question regarding the why was your conclusion that coverage should be improved.
Yes.
We also have a question.
Because,
yes, please, Marcia.
Yes, it's when we the vaccination schedule for these infants, and not most of them, but there is an incomplete variant of this age. So it's important with this this schedule will be full completed. It's based in our results, and not for our results, but with the others, mainly the importance of these vaccines against target and non-target diseases.
And also we have a question from Paulo Videira asking if you preview that in the future patient microbiological characterization could anticipate OPV or any live attenuated vaccine?
No, I know I don't know if I understand the question.
Paula, would you like to clarify? I believe Paula is asking whether you should characterize children's
before vaccination.
Yes.
Do you think we should take into consideration the microbiome or the before vaccination?
Yeah. Mm-hmm. Yes, it's, for example, it's in low and middle income countries, it's known that oral vaccines, there is not the same effect and for children in high income countries. And it's known that for example, successive episodes of diarrhea can compromise the gut microbiota, induces some degree of intestinal dysbiosis. And this is bad or not good for the effect of these vaccines, because they compromises not the local immune system for other bacteria that can compromise the effect of these vaccines there. So it's important, I think it's for these oral vaccines, I think it's important to characterize, to review if the effect will be a total or compromised by this dysbiosis and this intestinal dysregulation.
Thank you, Marcia. We now have some questions to Irina. The participants can all, of course, still ask some questions to Marcia, but we'll move now to Irina now. One question here is, you mentioned that this Legionella has a new organization. Could you specify a little bit more? What would you describe as the major difference at the level of all genome? And also whether this Legionella also acquires genes from human host cells?
Mm-hmm. Okay, regarding the first question, so analyzing the genome of this new strain, it was found that it had a kind of a chimeric backbone. So regarding, in terms of serogroup, this outbreak strain is very close to serogroup 1 strains, which are many clinically relevant strains, such as the Lutonella Pneumophila peris that I've taught. But phylogenetically, it's closer to other Lutonella Pneumophila subspecies, which is not from serogroup 1. So it seems to have acquired genetic material from phylogenetically distant strains. In terms of what it changed in the genome, a couple of things. So it seems this new outbreak strain has an secretion system, which is not present in the well-known Legionella Pneumophila clinical strains. It seems to have other virulence-associated genes, such as efflux pumps. But as far as I know, none of these have been functionally analyzed. So it's just in terms of prediction in the genome. And the other question was?
Yeah, so you've already addressed this one, or what do you think it's the main impacts in pathogenesis of Legionella of the localization of this effector in nucleus? I don't know if you'd like to add something else. Also, sorry, just related with this, it wasn't entirely clear to me whether there is some evidence that this can be related with the person-to-person transmission case that has been described, or whether this is just an hypothesis?
It's possible. It has virulence-associated genes that are not normally present in the well-known clinical strains. So it's possible that this acquisition of new genetic material from different subspecies of Legionella has enabled it to acquire more virulence traits than the regular well-known strains. Regarding the other question about I think you asked something about the microbiome in the lung, if
the question was whether Legionella also acquires genes from human host cells.
Okay, so it's not known. There is a very recent study that shows that during infection with Legionella, there are also other bacteria that may be present in the lung, and even amoebae, although it's thought that amoebae come from the aerosols in which Legionella come to. So it's, yes, I think it's in theory possible that Legionella is also able to acquire genetic material in the lung, but there are no studies so far about that. Regarding the impact,
the pathogenesis, sorry, I distracted you with the person-to-person transmission, but in regards to the pathogenesis or the severity, or are there any thoughts on that?
Well, so far, what we know is it has many or several different genes than other clinical strains of Legionella. So yes, it may enable this strain to be more pathogenic or more virulent. Regarding this effector that I mentioned today, the VFX protein, what it's doing in the nucleus, yes, we don't know yet. There are a couple of Legionella effectors that have been found to be targeted to the nucleus, and there they affect transcription of the eukaryotes, mostly in pathways related to immune signaling. They do so by modifying histones, so leading to increase or decrease in transcription of host genes. We don't know yet to which molecules or proteins or DNA it may be binding in the host cell, but that's our next question and the main goal in the near future to find them.
Thank you very much, Irina. I believe you fully addressed all the questions from our participants. If our participants still have any further questions, we can then move the discussion to our network chat. Thank you very much to our flash presenters. And now we move on to the next session, which will be moderated by Maria. Thank you very much.
Thank you, Andrea. And hi, everyone. I'm Maria Grazia Rossi. I'm invited assistant professor and researcher at the Faculty of Social and Human Science at the NOVA University of Lisbon. I'll chair the last two blocks of flash presentations that will include 3 talks. As you are going to see in the next 2 talks, we are going to talk on microbiome by discussing research coming from the areas of public health and communication. So I think I can invite the first speaker of this block, Miguel Costa. Miguel, are you there?
Yes, just a second, I will share the screen.
Okay, so while Miguel starts sharing his screen, I can introduce him. Miguel Costa is PhD candidate of the International Global Public Health Doctoral Program based at the NOVA National School of Public Health. And he is going to give a talk on microbiome and public health opportunities and challenges. So just let me remind you, you have 10 minutes to discuss your work.
Okay. Good morning. My name is Miguel Costa. I'm a PhD student at National School of Public Health. I'll present a review of challenges and opportunities associated with microbiome science in public health. I'll start by presenting some basic concepts of public health, the microbiome, and talk about microbiome determinants as an introduction to the topic, and then address applications and opportunities in public health, research challenges, and we'll conclude with some key messages. Public health is the art and science of preventing disease, prolonging life, and promoting health using organized efforts of society. As already mentioned by the colleagues, microbiome is a specific. It's asking to start my video. I'm sorry?
Yes, your video disappeared. So if you want, you can share it again. Now, okay.
Okay.
We can see you now.
Microbiome is a specific community that occupies a well-defined habitat with distinct physiochemical properties. The term not only refers to the microorganisms, but also encompasses their theory of activity. It is a complex host microsystem that co-evolved over time and is mostly shaped by environmental and dietary conditions. Here I have some examples of microbiome determinants. There are different factors that determine the early construction of the microbiome. such as the in utero colonization, the environment at birth. An example of the environment at birth can be the mode of delivery, such as cesarean, that will determine the microbes that will colonize the skin of the baby. Also the mother's milk that help to modulate the immune system and can select specific bacteria. Other determinants are determinants that will change over time, that will change over the human lifespan. Some of these determinants are lifestyle, that can be, for instance, a sedentary lifestyle, the environment or surroundings, diet, that can be the use of spices, for instance, from particular cultural practices, and other determinants such as age, disease, and antimicrobials. From all these determinants, there are some that seem to be more important, such as the environment, diet and malnutrition, antimicrobial therapy. The environment can be because of our surroundings will influence our microbiota. The diet, because what we eat shapes which microbes will grow and blossom. The antimicrobial therapy, because those of antimicrobials can damage the microbiota and decrease its diversity. Some examples for environmental determinants can be divided in macroenvironmental factors and microenvironmental factors. For macroenvironmental factors, we can have pollution, socioeconomic factors, such as the reduced access to medical and dental care, the built environment, including structures such as our homes, workplaces, schools, and green places, and microenvironmental factors like smoking or alcohol consumption. For diet, an unhealthy diet can result from the consumption of fast and processed foods. As we know, good food selects good bacteria. So outcomes of eating poorly may influence the nutrients that we need to stay healthy. And for antimicrobial therapy, the overuse of broad-spectrum antimicrobials and the increased development of antimicrobial resistance. Overall, these determinants can influence metabolic functions, nutrient synthesis, and the immune system. It's also important to mention that human resident microbes are not equally good, nor is greater microbial diversity. It's not a one-way street. The microbes interact in communities and respond to their surroundings. Some examples of applications in public health are the ones that are associated with genetic epidemiology. I'm sorry, and some examples are prognostic for early stage disease screening that can be used as a complement for cancer immunotherapy, or to predict treatment response or study microbiome drug interactions. For population scale studies to observe associations of phenotypes or exposures with microbiome features. Some examples of exposures of interest can be the determinants, the microbiome determinants, such as environmental and dietary determinants. Other applications are chronic disease diagnosis, management, and risk assessment, infectious disease monitoring, diagnosis and prevention, Some preventive and therapy strategies may include the use of lifestyle modulators, such as prebiotics and the change of diet, but also probiotics to stimulate the microbiota, diversity, and also to stimulate the immune system. Some examples of probiotics can be smart probiotics to incorporate or select specific bacteria. Some opportunities in public health are to study microbiome disorders to improve management of diseases. Here we can look at those of antimicrobials. For instance, when we get antibiotics in the hospital, it is expected to treat or prevent infection, but by doing so, the antibiotic therapy can increase the odds of curing a hospital infection. Dietary modulation to introduce compounds that microbiota needs to induce a specific response that will benefit the host or treat the health issue in question, and understand the role of macro and microenvironmental factors that can offer insights on how to prevent chronic and uncommunicable diseases. There are many challenges to research in public health. Some of these are the risk of bringing together causation and correlation, as the microbiome can influence and respond to many phenotypic and environmental factors. The sensitivity to technical bias, contamination, and measurement errors due to a diversity of study methods and assays. The inter-individual variability of the microbiome that may result in different protocols of samples, of sample collection in the same study, difficulty in data generation methods, and reproducibility of results. And to conclude, some key messages. Challenges in public health determine microbiome, but microbiome also provides opportunities in public health. Methodological challenges need to be solved to better define and develop public health opportunities. Microbiome disorder can be the cause or the outcome of disease, and a different way to see infectious diseases, not just an interaction between host and pathogen, but also including microbiota. Thank you very much.
Thank you, Miguel, for your interesting presentation. As you know, we will have the discussion after the next presentation. So please, if you have comments or questions for Miguel, use the chat. So I'm about to give the floor to Claudia Pernencard. She is researcher at the NOVA Faculty of Social and Human Sciences and invited professor at the School of Fine Arts and Design, Polytechnic of Leiria. She is going to talk about ontology and semantic for an embodied conversational agent, a health value for inflammatory bowel disease patient care. So, yes.
I'm just going.
Yes, I have just to give me a moment. Okay.
Okay.
Now I think you can show.
Yes. Just
welcome.
Going to okay. Thank you so much, Maria. I'm going to share my screen. Okay.
Yes, everything.
It's perfect. Perfect.
You have 10 minutes.
Okay, thank you so much. Good morning, everyone. I would like to thank you to the committee of the conference for considering social science and humanities as a relevant topic to be included in this conference. It's always a challenge. I'm a researcher also in digital health. And today I'm going to present this exploratory research ontology and semantic for embodied conversational agent, a health value for ABD patient care. ABD highest prevalence rate, according to a study from Lancet Gastroenterology and Hepatology from 2017, shows that 6 million cases globally exists. Considering that we have two countries, two main areas, which is USA and UK, with high level of these incidents, we believe that there is different opportunities for several areas, scientific areas, to be more assessed related to technology to help ABD patients. So just a quick summary for those who don't know exactly what does it mean, a conversational agent. Conversational agents is a common chat, what we call a chatbot. So this is a digital tool that exists currently in our mobile phones, for example, that use behind machine learning and artificial intelligence methods to orientate a conversation between a robot and a user. And this, we can find these tools in some services that exist in our mobile phones applications. The challenge here that we find is just to understand what does it mean a semantic ontologist related with a chatbot. Semantic in communication science study the meaning. So when we join technology to communication science means that we need to understand how computers interpret with accuracy what patients are saying in their dialogues. So this is what we have here related with ABD. So our research topic has an exploratory study, is to understand if a conversational agent could reinforce the health value for ABD patient care. So we intend to understand mainly if a bot, for example, in a mobile phone of a patient could help and contribute for has a health value for ABD patient care, promoting their quality of life. So we have other research questions. How do academics describes the use of chatbot by healthcare professionals and also ABD patients? What are the implications that exist using when we are talking implications means the rights of the patients, the informations that are available in our mobile phones, our GPs and something like that, that we are using chatbots in the healthcare of ABD and healthcare professionals. Other point that we want to understand is if the patients' profiles that exist currently in Portugal are target or are able to use this technology with accuracy in their mobile phones. This is our main research questions. And we started this question with a strategy that includes a systematic review, because we needed to understand first, before starting the project, we need to understand a little bit more the context and how the academics and how the world is seeing introduce a chatbot in ABD context. So it was very interesting, this part of the research, because we conducted a systematic review that included a search in different databases, and our search was ranged between 2015 and 2020. We defined 80 keywords, and this query was included in this all databases. We conducted this systematic review with the researchers, 3 researchers from different fields, okay, from communication science, from design, and from healthcare, including a Portuguese public hospital. The screening, it was interesting and open. It was an open opportunities for all team, because we concluded with screening the screening numbers that you can see, that we find 7,586 articles, but only 9 after 3 cycles of reviewing, always double reviewing. It was interesting because only 9 studies, okay, was following the well-defined criteria. Okay, we define a structured criteria. We use a Parsifal electronic tool, which is Parsifal, to define and resist our protocol studies. And after that, these 9 studies, we decided to create a literature map to identify the relevant details of each article for us to understand how should we conduct our study. And we have here some interesting points about a few literatures reviews that we found that, for example, machine learning has a technology. It's not new in gastroenterologists. They are using since 10 years ago, but in particular for imaging. It's very interesting because in chatbot, it's not so well defined. For example, the way patients see at this moment these conversational agents, they understand that, no, this is mobile phones. No, this is not a mobile phone application. This is a tool that exists inside of a mobile phone application. And this is very interesting that literature reveal to us. And also an interesting project regarding mental health where chatbots are being used in suicide preventions and cognitive behavioral therapy. This study also show us that that is a challenge regarding the dialogues about the patients, what people said through dialogues in, for example, a conversation between a robot and a patient. And mental health, it's like a large area that are very complicated to define. Some query regarding some specific semantics that bot can talk with this kind of patients. So this is a challenge also to cognitive diseases, and we found amazing this study, and it was an inspiration for us, because we only found one study that explored the use of chatbot regarding ABD patients. And this studies, it was developed in 2019, between 18 and 19, from UCLA, Crohn's disease, ABD Crohn's disease center, and it exists only article published from this team and a poster. So this is very, very interesting and reveals a lot of opportunities for our team. And the results from these articles was very interesting, because they search they extract from UCLA ABD database a lot of lines dialogue. Okay, this was from the database. They didn't specify where they came from, because I believe that they cannot tell that in this exactly this article. But they filter 424 patients were studied with 324 message. So it's a lot of message to extract some lines. and they define a relevant strategy, which was, we're going just to study the first 500 lines of each dialogue, because we believe that is in the beginning that most relevant content are effective. Okay, so they define these strategies. Okay, it's for us, we don't know yet if it's right or if it's wrong, but there is a curiosity in these particular strategies, which is they define it also 8 categories by their frequencies. Okay, and the first 2 categories with more percentage related with what these patients were talking about, what was medications and communications, and for us was was relevant, because we thought, okay, maybe symptoms, maybe it's appointments. No, it's about that medications that this sample are talking about, and communications. So medications, it's a well-known, for we have conducted other studies regarding social media, how ABD patients discussed several subjects regarding social media, and we know that, for example, medications, symptoms, are hot topics regarding what patients discussed in private groups in social media. So communications, it's a little bit outsider from our perspective as a research team. But medications, it's included. We cannot put outside that these samples, it's only dialogued USA based. So I believe that outside of USA based, the dialogue samples also change it. Okay, we believe that. And other categories are laboratory search. Symptoms comes on the third, fourth places. So for these patients, symptoms maybe are outside of the what we are talking of their priorities. I believe that not, but maybe medications, it's more important for them. Appointments seems to be after. So for us, we thought, okay, symptoms and appointments for these sample groups are not so relevant. Maybe there are some issues that we can consider when we started our research in Portugal with ABD, with Portuguese ABD patients, that we can consider it also. Procedures and financial and insurance, we are talking about, for example, USA population, financial and insurance. It's not a problem for them. We thought, okay, maybe because we are talking about a private hospital.
Claudia, sorry to interrupt you. Just if you want to leave space to the discussion, I ask you to go to conclusion. Conclude.
Just 2 minutes. So the discussion told us that was not clear enough about which are the best strategies to be adopted regarding ontologies that can vary. Okay, because it exists only one study that can help researchers how they can create this ABD ontologies, and it USA based, and electronic medical records also difference in certain aspects, as for example patient's behavior and physicians' experience from clinic to clinics. So we concluded that open language to characterize still a challenge. ABD ontology, it's not stronger enough regarding some guidelines to help researchers to find a concrete way to create a specific ontology for a chronic disease. ABD patient care shows the promises, and the chatbot, maybe it's not for health professional so relevant. They have all other issues to to talk about it, to discuss about it. So our future work is involving a PDI, Portuguese Association of the Disease and Value for Health Collab, which is a collab from NOVA, to create some steps regarding our project, which is to create an holistic model first, to outline statistical data using with the ABD patients collaborations from PDE associations. After that, we intend to screen the data and their dialogues to create this a sample of ABD ontologies, and finally a small prototype with a concept proof. We are to researchers from different areas. We have here in our team Ana Sampaio, which is chairman of APDI, Ana Rita Londral, also from Collab, VOCollab, executive director also, and we have a mission. Me and Ana Sampaio, we have ABD diseases, so it's important to defend to give voice to the patient through research. And Rita Londral can help us with their interdisciplinary team, considering their work with linguistic expertise. And we have a challenge. How does the stakeholders comprehend the value-based healthcare, putting the patients in the front and not the stakeholders? Thank you so much for your time.
Thank you, Claudia. I invite Miguel also to activate the video. Thank you, Miguel. So I think that now we can start the discussion. As you can see, we have many questions in the chat already, and you can continue to ask other questions or comment on these two presentations if you want. I think we are going to start with Miguel. There are at least two questions for you. I'm going to read the first one that is from Ligia Saraiva. So she asked if in your group, what are you doing to implement these public health measures related with the microbiome?
This area is still in its early stage. Right now, we are working in the study of antimicrobial resistance, but not yet specifically related with the microbiome. It will be something to further develop in the future.
And there is also another question about the future of microbiome research in public health. That the question said that it's clear that this research is at very early stages. So what would you highlight as the next steps in the development of this research area and the corresponding applications? Which areas are more promising in your opinion?
More promising, perhaps the applications that can be used for diagnostics to look at microbiota's health, the individual's lifestyle, environmental factors that may help to identify health issues or what needs to be reinforced in our body. But there is a need for more evidence to develop and implement appropriate measures at the population level. As everyone microbiome is different, also each individual needs are also different.
So I also have a question myself for you, Miguel. I was thinking when you talked about the different challenges that you have in this field when you design microbiome interventions to target community and public health challenges. And I was thinking to the ethical dimension as a possible further challenges. Do you have any thought on this dimension? dimension are you thinking to take into consideration also this dimension in your future research and studies?
There are some ethical issues in those of, for instance, the fortified food products at the population level, but I don't have a very good, I don't have an opinion, a good opinion format to give still in this matter.
Thank you, Miguel. This means also that there is space, of course, for new collaboration and new research in this field. So I would like to move now and ask questions to Claudia. So we have many questions for you in the chat. So the first one from Andrea Leite is how is the health value measured in your study? Is it in a value-based healthcare perspective? Are prompts considered for example? So are patient-reported outcome measures considered?
So first, we are trying to understand if this will be a value. We believe that this will be a value-based healthcare perspective. Okay, we believe that, but in the meanwhile, we can consider it prompts, for example. Yeah, we can consider Andrea. But from the first, our first step, it's to base this value based in the patient perspective. Okay, this is only our first step.
Thank you. And also, it's always a question from Andrea Leite that we're asking is this approach that seems to be particularly relevant for ABD patients could be also applicable also to other chronic disease patients.
So ABD patients, what we have here is chatbots communicate using a specific semantics to understand what the dialogues between robot and patients. So if we change the disease, for other chronic disease, I believe from what we have searched until right now, that some aspects are going to change. So we cannot use like a formula or a library to change to other chronic disease. Okay, we are thinking about also if it's possible to have disease with same behaviors, patients' behaviors, and with that create a particular pattern to apply to other chronic diseases. To similar.
Yes, thank you, Claudia. I just was looking that you are still sharing your screen, so give me a moment. Okay, so that we can see each other better. No, it's okay. And so another question from Claudia Conceição this time. So it's not a question that addresses the main objective of the presentation. It's more like a curiosity. So Claudia would like to know if topics as use of prebiotics emerge in patients' communication. I think in the context of the chronic disease you were talking about.
Yeah, exists, Claudia, and it's very interesting because I have been studying with a PDE involvement, this communications in social media, and the probiotics. It's a hot topic in patients' communications, but in a simplest way. It's not a scientific way, because patients are always always involved how they can be better, how they can have a better quality of life. But they are not worried about the scientific main, because they cannot understand, most of them cannot understand what is behind of the benefits of using, for example, probiotics. But it's a hot topic. It's more like brands of probiotics that they discussed in these groups.
So if you still have some questions for Miguel, we still have some time for the discussion. In the meantime, I have a question on myself for Claudia, and it's a very general question that is related, it's on the connection between your study and the broad area of patient-centered medicine and also personalized medicine, because patient-centered medicine is also based on tailored communication and messages. So I was wondering how can you tailor messages by using conversational agents? How do you do if you can do it?
Depends of this is a very critical point, because communication using robot depends very points. Two main points. First, it depends of digital literacy, how far digital literacy of the peoples goes. And the other part is health literacy for the peoples who are interacting with these spots. Because from previous studies regarding other fields that I'm involved, we understood that health literacy also it's a key point in the digital world. So, for example, when we are talking about some semantic language that it's like a sample that people are used to interact with the bot using other services, they understand and they reply exactly what the meaning from the other examples. But sometimes it's not the same. Okay, and it's why it's a challenge considering the chronic disease, because we cannot use a pattern from different other areas to chronic disease, to the chronic disease.
So from what you are saying, it seems also that the educational profile of the population is very important.
Very.
And related to this communication with patients, one question could be, which are the hot topics in the data you analyzed and you are analyzing?
Sorry, I didn't understand your question.
About topics that you are analyzing in this communication.
Okay, so we are analyzing first the baseline, what we call the baseline of the linguistic. So how these patients going to interpret if they understand that some chatbot is interacting. First, the patients needs to be involved, to be engaged with the robot, and it's very important for them to understand that the bot is there to help them. It's not like other layer of complication in that disease. So first, this is a challenge. And regarding the semantic ontologies, we are just trying to see if, for example, a simple phrase which is "Hello, how are you today?" How this is so large to the people who are suffering currently with the chronic disease, it could be a good way for them to be involved with the therapy, because we know that most of the people don't follow their therapy well. So this is just a small example, considering your question, that we are just researching.
Thank you, Claudia, and thank you, Miguel, for this nice discussion. I think that now it's time to move on the and to invite Pedro Lopez for the last flash presentation for today. And it's with great pleasure that I'm going to introduce Pedro Lopez. Pedro Lopez works as a general practitioner at Hospital da Luz, and he is medical lead at Pfizer's Hospital Business Unit. The title of his talk is Antimicrobial Stewardship Programs and its Impact on Microbiota and Health. Thank you for being here.
Good morning, good afternoon. Let me start by sharing my screen. Thanks for the nice introduction. As was told, my name is Pedro Lopez. I'm the medical lead for Hospital Business Unit in Pfizer, Portugal. I'm a physician by training. And on behalf of Pfizer, I'd like to thank the organizing committee for the invitation and for giving us room to talk about such an interesting topic and emerging topic, because antimicrobial resistances and also antimicrobial stewardship is clearly on top of mind of all of us, and it should be even more to reduce the impact that this public health problem will have in the future. And I would like to start by sharing with you a statement from the Director General of the World Health Organization. That is, as we gather more evidence, we see more and more worryingly how fast we are losing critically important antimicrobial medicines all over the world. And this is critically important because we know that we don't have that many new medicines coming addressing the unmet needs that we have, and also due to the resistant topic, and it's where I would like to start. We know that antimicrobial resistance is one of the biggest problems in terms of public health, and it's challenging. It's not just a challenge for the human medicine, but also to the animal medicine and the environmental health. And this is led by the overuse and misuse of antibiotics in general. And I believe, and I will reach that point a little bit later, that the issue becomes even more important with the COVID pandemics, because it increased the bad practices, and also it's likely to contribute to an antimicrobial resistance increase. This is not only a problem of health, it's also a problem of financial sustainability, a problem of global health. And when assessing all the initiatives that we can put in place to address this problem, we should evaluate the impact of these initiatives also from a health perspective, but broader in considering all these other phases of the same problem. The antimicrobial resistance also led to several adverse consequences, and we can see here a list of them: severe illness, prolonged hospital admissions, the healthcare costs that are higher, the burden in terms of public health, but also, as I said, sharing the statement from the World Health Organization General Director, also because we are reaching a point in time where we don't have much options to lose treatment weapons. So having more treatment failures and somehow jeopardizing the ones that we have can be an issue in the near future. We know that, as I've said, the number of antibiotics coming to the market has declined very, very critically. And we are facing a loss of effectiveness also due to the antimicrobial resistance. The number of people dying from AMR is quite high, 700,000 per year. But if we don't do anything to overcome this problem, by 2050, the forecast is that we will have only in the European Union 33,000 people dying from infections due to resistant bacterial strains. Sorry. as I've said, COVID-19 didn't bring good things, and we know directly, and we are still living the issues that and all the impact that COVID bring to our lives and to our community and society. But also in terms of AMR, it was not a good situation. Mainly because there are a huge number of patients that were treated with antibiotics, even if they don't have any condition that is supposed to be treated with antibiotics. Patients with mild disease, without pneumonia, or even with moderate disease were taking antibiotics, and there's no formal recommendation towards that. So the pandemics also is a threat to the antimicrobial stewardship, and also contributing to AMR increase. And several studies have been done to observe and to document the way these patients were treated. As you can see here, 72% of the COVID-19 hospitalized patients in some publications received broad-spectrum antimicrobial therapies, which is even more critical in terms of the impact that may brings to the AMR field. And now moving to the antimicrobial stewardship, as this is one of the most relevant tasks that we have for the future. And I think we have still a huge room to improve the way we implement these kind of initiatives. And it's clear that we need to adapt the way we look to this, because people are changing, behaviors are changing, but not at the speed that we need. So this organization and healthcare system approaches are important to reduce the impact of AMR, but also to contribute to the best use of antimicrobials. And it's also critical as a community to engage the most broader audience of people that we can, because it's not only a problem of physicians or veterinaries or microbiologists, it's a problem that will affect all of us in the future. So it's really important that we raise awareness to this, and also that we understand where we are, and improve the way we monitor the impact on microbiota and also in health of antimicrobial stewardship. I was reviewing a couple of papers, and all of them conclude that the information that we have and the way we monitor these initiatives is not the best way, because they are not conclusive. So we should rethink, and also rethink the way we implement the initiatives, and rethink the way we monitor these initiatives, because in the end, for the government and for the payers, it's a matter of price. And if we do something that spends money, we need to prove that in the end, in the medium and long term, we will for sure have a return on that investment that is more than the investment in our individual health. There's a lot of things that we can consider when we think about antimicrobial stewardship. We have here on the table some types of interventions. Education is clearly one of the most important, and we were discussing about health literacy, and Pfizer has a strong commitment in terms of health literacy to the population, but also to the healthcare professionals. It's important that all the layers of the health system is informed about this topic and the importance of the antimicrobial resistances, and also to promote the implementation of guidelines and of local committees that can evaluate the practices regarding antibiotics prescriptions. And this led us to the formulary restrictions that can be a strategy also to agilize and to guarantee that we have the best practices in place. Switching from intravenous to oral, try to replace antibiotics, the antibiotic timeout, the antimicrobial cycling. There's a lot of strategies we can use and implement according to the local environment, and can be useful in terms of antimicrobial stewardship. And this is These type of interventions are not a sequence. They can be done at the same time, but we need to evolve and to continuously evaluate, and also to establish a mitigation plan to improve the outcomes, to improve the quality of our antibiotics use. We can see the cost saving in several studies published over time, but we can and we need to see more than the costs, because it's not just a matter of saving costs with antibiotics, it's also to understand the way we fight the infectious disease, and also what can we earn in terms of antimicrobial resistance, but also in terms of the impact that this can have in our health in general. We know, sorry? Oh, okay. Antibiotics can cause dysbiosis, we know that, this topic is critically important, because nowadays we know very clearly that dysbiosis is clearly linked with several health conditions, and it became a very important target in terms of therapeutics. So starting to implement antimicrobial stewardships, it's also important in terms of our general health due to this topic in particular. We know that microbiota diversity is something really relevant in terms of our health, and if we analyze, and there's several publications on this, if we analyze some healthy controls and compare that with specific populations with some diseases, we find that the microbiota is changed, and these changes shows some kind of pattern. So we can see that the changes induced by our behaviors since we are born in our microbiota can trigger in the future some of our most important health conditions, and we can see here diabetes, but other diseases as well. Gut microbiome is a very trendy topic, because it became part of our vocabulary, and also in terms of population, it's growing awareness and recognition, and it is really important that we continue to communicate the importance of the health of our gut microbiome, but we need to bring this and to link this with the optimal use of antimicrobial agents. Otherwise, we will be continuously harming our microbiome and contribute to the development of resistances. We know that there are intrinsic and extrinsic factors that influence our microbiome, and this is something that some of them it's hard to interfere with, but there are some that depends on us, and we can do something as scientists, as physicians, to try to recover from the harm that we have induced in this particular. The exposure to antimicrobials reduce the microbial diversity in our gut, and this is something very well established. And the lack of diversity and the changes in microbiome are considered nowadays as an amplifier of antimicrobial resistances. So it's really important to assess this, and also to reinforce our behaviors as prescribers of antimicrobials in the daily practice, to avoid moving this to next generations. And it's one of the biggest and the most consensual messages across the publications on this topic. Another point I would like to highlight is that, and is something that will have impact in terms of antimicrobial stewardship, is that we need to try to be the most strict as we can when prescribing an antibiotic. We know that the antimicrobials sometimes are not very target specific, so they have side effects in terms of bacterial diversity, but we need to stimulate the physicians and all the the antimicrobials prescribers to have this in mind when prescribing. We have several evidence showing that shorter courses of antibiotics may have benefits in terms of long-term when talking about gut microbiome, and also it's important that we prescribe narrow-spectrum antimicrobial agents on top of the shortest time. There's a lot of other measures that are suggested in the literature without very robust evidence, but they are important that we have that in mind, trying to prescribe more and having in consideration the route of administration, but also the metabolism and expression of the drugs. We know that the oral antibiotics are more prone to induce changes in microbiome. So it's probably a good practice to bring that to our minds when prescribing. But of course, we need to go step by step and try to establish these measures in the long term. In conclusion, I think we are clear that the wrong and the misuse of antibiotics is leading to several changes in our gut microbiota, and also these have a huge impact in our profile of antimicrobial resistances, and the microbiome also plays an important role on that part. We need to change and to challenge prescribers to change their behaviors. And this is something we need to do in a continuous way, but also to start monitoring it in a more effective way, because otherwise no conclusions will be taken from the interventions we are doing, and the commitment and the engagement of the government will be lower for the future. Even if all the scientific community is aligned with this goal, I think we need to generate more evidence that demonstrates that we will be saving our health, but also saving in terms of costs in what regards to antimicrobials use. But also, I would like to conclude by highlighting that all these approaches that target gut microbiome are of a huge impact in our individual health as well. So it's something that we should start at home in our own practices. So maybe we should start targeting not only the healthcare professionals, but also the young kids, the children at school. And we had several good examples in the past with ecology and recycling. So we should bring these principles also to antimicrobial stewardship and try to understand how they can how that can influence the outcomes in the future on these regards. Thank you very much for your attention.
Thank you, Pedro, for your rich presentation. So now we have some time for the discussion, and you can use the chat for your comments or questions. So we already have a comment provided by Ligia Saraiva that she wrote a comment that say is we need to go back to basic knowledge on the function of pathogens to find new antimicrobials. Are the pharmaceutical companies willing to participate in these basic studies that usually will provide data that will be important only many years ahead?
Yeah, from a Pfizer perspective, we are clearly committed with this, and in a global scale, we are always committed to gather all the information we can to create a clear picture and ongoing characterization of our resistances in terms of antimicrobials, of course, and also to bring that to our development plans, also to develop additional information and probably develop new drugs that can address those needs. But this is a long-term run, so it's not something that we can have in a short period of time, and all the community should come together to identify, and we have several paths to that, because we are not just supporting our own investigation, we are always supporting the community to investigate new molecules, new mechanism of action, or at least new ways to fight against the antimicrobial resistances, and also to support antimicrobial stewardship. But it's clearly and unfortunately a field where there are no big news for a couple of years now. So we need new weapons, but we need more than that to use better the weapons that we have.
So thank you. We have two more questions for you. The first one is by Antonio Pedro Fonseca, that asked, how do you see anti-pathogenic approaches to minimize the use of antimicrobials in a bactericid or bacteriostatic approach?
It's a new approach, and I think it's something we could or we should try to evaluate. I think that's we need to evolve from the current options that we have and the current mechanisms that we are using to fight bacterial infections. So I don't know any specific drug under development with this mechanisms in specific, but it's something that it's a field for further development. So it's I don't have any specific insights, information to share, but it's a new field to explore.
And our last question for today, and it's from Claudia Conceição, that asked if you are aware or you know something about which are the last recommendations regarding antimicrobial prescriptions. So if there are guidelines, she mentioned given by medical association, for example, in Portugal or abroad.
There are several guidelines disease specific, but I think that very often physicians use guidelines as a general orientation, that they don't follow that. We observe very often a mimetization of behaviors. So, and we need to educate and to contribute to that. Medical education is critical, and we need to leverage from what we learn in the university desks. Usually some of us, and I speak as a physician too, we try to use the same drugs over time, and sometimes we use more our experience instead of the evidence, and that's the behavior we need to change. There's several guidelines, more international guidelines than local guidelines, but there's a lot of them, and it's something that we need to educate physicians to commit. And at the hospital setting, the commissions that are in place usually try to work closely with the physicians to improve the practices. And I believe that being this practice more and more often and more close to the field, it will have some results. So our commitment is also to contribute with our educational programs to create discussion forums and meetings to educate physicians to adopt the guidelines, but also to adapt the guidelines according to the local microbiology and environment. Sometimes there are differences between Portugal and Spain, or Portugal and Italy and other countries within Europe, not going further, but even in Portugal, there are differences between institutions, and that local adaptation is critical. And it's important that the local committees can take from the guidelines and also try to adapt to the local needs.
Thank you, Pedro. We have also a small comment in the chat that you can read. I think that it's a specification of the questions that you were already discussing. I don't know if you want to add something, or perhaps it's if it's about the recommendation you discussed on slide 15, if these recommendations are already translated in guidelines.
No, they are just thoughts and considerations from the several publications. They are not, there is no formal guideline in terms of antimicrobial stewardship, and there's only general principles that should be adapted to the local practices. And I think that is something that, as I said, we have to build up in a national level, but also at the local level, because antimicrobial stewardship is a local issue, I would say, is an individual issue, and we should have a cross-sectional approach to this to improve and to leverage the outcomes in the end. But it's a work that can be done, and the scientists and the medical associations can clearly work on that to establish at least a baseline to improve for the future.
Thank you again. Now I'll give the floor to Alessandra Fernandez that will give the announcement of the Best Poster Award.
Okay. Thank you, Maria. Okay, good morning, everyone. So we've been here listening to so many nice, very outstanding presentations, and now it's time for awarding. So I will present the Best Poster awarded, and I will try to say it now. So the awarded is Mariana Delgadinho with the poster Gut Microbiome of Angolan Children with Sickle Cell Disease. So if you were presential, we would get a very nice applause to Mariana. I don't know if she's here in the audience. I hope so. Someone from the poster, so maybe there is Mariana. Yeah, Mariana is here.
Yes, I'm
Hi.
here.
Hi, Mariana. Congratulations.
Thank
Very
you.
nice work. Can you just explain a little bit about your work?
Yes, I'm starting my PhD in Ciências Biomédicas in HMT, and it's about analysis of the gut microbiota in children, in Angolan children.
Okay, yes. Great work. We wish you, of course, all very nice success, your PhD, and with the work that for sure will increase our knowledge about the disease, and will impact, have a great impact not only on these children, but all over the world. So congratulations, Mariana.
Thank you.
Okay, so now I think we're almost ending, and to that job, I will leave Andrea again.
Thank you, Alessandra. So I have the job now to close and leave some concluding remarks. First of all, I would like to thank everyone for being here today, our participants, also all our presenters who have definitely contributed to an overview in regards to the research we have now, the cutting edge research we have now in this area, which seems really promising, but at the same time, with a lot of challenges ahead. So certainly this will be an area to continue to follow, and eventually to come back in a few years from now to update us on the developments in regards to microbiota and health. Also, I would like to thank all my fellow colleagues from the organizing committee for this organization, and also the remaining group for all our interesting discussions and work. Today, we've seen, as I mentioned, various areas of research, ranging from experimental, more lab-based research, till the observational studies that are being conducted, and also the potential applications at a more population level. And so I leave now these main ideas, and also I'd like to remind all the participants that they can go back to our website where all the posters are posted and available for future reference. And finally, we can continue our discussions through the network chat, which has been created through this application that you can download Telegram through our website. Thank you, everyone. It has been a great pleasure, and I hope to see you all in the near future.